So this leads to a large number of advantages for chloroplast transformation. So I'll take them from the left. Hyperexpression. The most expression you're going to see for a protein via nuclear transformation is about 0.1% of soluble leaf protein. I was talking to Ralph Bock at a meeting relatively recently, and he reckons he's had 79% of the total soluble protein in the leaf expressed as transgenic protein. So phenomenally high levels of expression are possible. Now, he said the plants weren't looking too well. It's hardly surprising when most of the effort is actually going into making a foreign protein which is sitting in the chloroplast. Multigene engineering is possible through these cistrons. Maternal inheritance. The chloroplasts and the mitochondria are eliminated during pollen grain formation. And what this means is that the chloroplasts and mitochondria must be provided by the maternal parent in a sexual cross. And what this means is that once you actually put the gene in, it's maternally inherited. It's only transmitted through the female parent. What this means is that a lot of people are worried about dissemination of pollen carrying transgenic transgenes. But you don't have to worry about this if you engineer the gene to the chloroplast, because you know that it's not going to make, the gene's not going to make it into the pollen. And this helps with gene containment. No pleiotropic effects. Like I say, when you put a gene into the nucleus, you don't know where it's going in, and you don't know what effects it's going to have in terms of interacting with other genes. Because the chloroplast genome is reasonably small, it's going to have relatively little interactions with other components. No position effects. We know exactly where these genes are going in because we put them in via homologous flanking sequences, so they go in by homologous recombination. So there's no problems about actually putting them in and they just happen to go in behind a strong promoter or a strong silencer. It's not going to happen with chloroplast transformation because we know exactly where they're going in. No gene silencing. One of the big problems with nuclear transformation that I haven't talked about up to now is gene silencing. If you have more than one copy of a gene being expressed by a particular promoter in a plant cell, then you tend to get less expression rather than more expression. This is counterintuitive, but it is a reality. So you would think that if you can put 50 copies of a gene into a particular cell, you would end up with 50 times as much protein as if you put one copy of a gene in. This is not the case. Something happens. There's some interaction between the different promoters such that rather than getting more gene expression, you get less. And this is the reason why Agrobacterium infection is so effective in development of transgenic nuclear transformants, because Agrobacterium transformation tends to put in single copies of your gene. Now, Agrobacterium transformation is no good at all for plastid transformation. When you put DNA into a cell and are trying to target it to chloroplasts, you need lots and lots of DNA, lots and lots of copies. So Agrobacterium transformation is not going to work for plastid transformation, whereas it is very highly effective for nuclear transformation. Partly because of this, for nuclear transformation, Agrobacterium transformation means only a single copy put in and no gene silencing. No vector sequences. One of the problems with transformation is that when you do an Agrobacterium transformation, you have to have quite a lot of vector sequences associated with the Agrobacterium. And this is not the case with chloroplast transformation, where you're developing quite small plasmids which are replicated in E. coli. Okay, so let's have a look at plastids. There are a whole load of different types of plastids. Chromoplasts, chloroplasts, the ones we were most familiar with, amyloplasts, which are used for storing starch, leucoplasts, elaioplasts, etioplasts, which are contained in etiolated plants. And whether we can successfully get plastid transformation depends a lot on the type of plastids that are contained in your tissue. When you're doing a transformation initially, when you're getting the DNA into the cell and trying to get that DNA into a plastid, it helps if you've got a lot of plastids to put that DNA into. So you want to look at this list here from chromoplasts down to etioplasts and say, well, which one of these has got lots of chloroplasts in each cell? And the one that does is the chloroplast. So chloroplasts may contain 100, so each leaf cell may contain 100 chloroplasts, and each chloroplast may have 10 to 14 nucleoids. Nucleoids are where the plastid DNA is actually attached to chloroplast membranes. So there are several copies of plastid DNA per nucleoid. So when you're transforming, when you're trying to get the DNA initially into the cells in order to transform, you want lots of plastid DNA there to facilitate the recombination. So the ideal tissue is chloroplasts. Got lots of chloroplasts per cell, lots of nucleoids per chloroplast, and several copies of plastid DNA per nucleoid. Now, when you're selecting to try and stabilize the gene expression, then you don't want lots of copies of plastid DNA in each cell. So you want to try and minimize the number of copies of plastid DNA in each cell, and the way to do that is to actually to go through proplastids, which are typically found in meristematic cells. So proplastids would contain only 10 to, each meristematic cell would contain only 10 to 14 proplastids, each only carrying 1 to 2 nucleoids per proplastid. So basically you're introducing a bottleneck by going through meristematic cells containing proplastids, where it's relatively easy to get rid of the non-transformed copies of plastid DNA. So initial transformation with green tissues with chloroplasts, selection using meristematic tissues containing proplastids. The plastid DNA is a kind of a funny kind of figure of 8. So you've got a large single repeat, a large single copy region at the top. Then you've got these inverted repeats, which is actually at the center of the 8, where the DNA sequence is actually repeated. And then you've got a small loop at the bottom, which is the small single copy region. And you can put plastid DNA or target your genes to any of these regions. When you go through the literature and start looking at where people have put genes into the chloroplast genome, you'll find that they tend to go for the inverted repeat. And the reason for this is that if you put a single copy into one of the inverted repeats, then very quickly it tends to be copied over to the other of the inverted repeats. So you end up with 2 copies of the gene in each plastid DNA, rather than just a single copy. So if you put it into the large single copy region, you can end up with a single copy of your gene. Put it into the small single copy region, you get 1 copy of your gene. Put it into the inverted repeat, and fairly quickly that gene will be doubled up. So it will appear in each of these inverted repeats. So there appears to be a reasonably high level of conservation of the genes and of the structure of the chloroplast DNA in higher plants. So Marcantia, in the middle there, is a liverwort, and you're comparing that to rice, which is a monocot, maize, which is a monocot, and tobacco, which is dicotyledonous plant. And there are a large number of genes which are regulated in the chloroplast. So these are genes associated with the genetic system, like chloroplast RNA genes, ribosomal RNA, transfer RNA, protein synthesis, photosynthesis, some respiratory proteins, and a number of other genes. And you can see that one of these is the RBCL gene, rubisco large subunit. Rubisco, ribulose biphosphate carboxylase oxygenase, is the most abundant protein in nature. This is the protein which combines carbon dioxide and brings it into organic combination.